Hello everyone, so this is Professor Scam clocking in to talk about Mendelian genetics, part uno. Before we begin, I have to yet again apologize for the crazy cat named Pharaoh that is tearing around my apartment. As you probably just heard a few moments ago, he has his squeaky mouse and he won't stop playing with it. I try to put him in my room, but he cried and it broke my heart. So and I apologize. He has to make an appearance in every video, so say hello to Figaro. Anyway, so Mendelian genetics, we'll be focusing largely on the first and second laws of Mendelian genetics, talking about monohybrid and dihybrid crosses and how they kind of relate to those laws. These are just the class goals for the genetics class that I teach. Alrighty, so when we're talking about Mendelian genetics, we have to, of course, talk about Gregor Mendel, right? So Gregor Mendel is the father of genetics. His life spanned between the years of 1822 to 1884. He was a monk in an Augustinian monastery in a region of today's Czech Republic. And he devoted much of his life to the study of inheritance of traits. Now we all know that the inheritance of traits are a characteristic that's possessed by an individual, many of which are heritable. They're what make us phenotypically human. So Mendel conducted his research, some fantastic research. And the amazing part about it is that he had no knowledge of chromosome structure or of meiosis or any modern concept of a gene, and yet he was still able to construct this monumental research. And his approach was strongly influenced by his training in both botany, which is the study of plants, and physics. And that physics component is what gave him this mathematical approach. So when we're talking about Mendel, he had a different approach to his research than what the standard norm was at that time. So the first thing was that he conducted what's called hypothesis-driven research. He formulated a hypothesis, and he also implemented the scientific method, meaning he followed these specific steps into implementing an experiment, forming a hypothesis, forming these questions, and then conducting the experiment to obtain results, and then discussing the results. And this all allowed him to have this well-formed experimental approach. So in addition to having that kind of strong foundation, he also had an excellent model organism that he chose to work with. He chose to work with the common pea, which we'll talk about in a little bit. But this is a model organism. He used controlled crosses. He had true breeding strains, and he started the experiments with those. So you started each experiment with a true population or a pure breed, per se. The model organism he worked with had binary traits. So it's either yes or no. It's one or two, yellow or green, etc. He quantified the results. He replicated all of his studies, and he used what are called test crosses. And he had a large sample size. So because he had a large sample size, he was able to replicate. The more you replicate, the more valid or substantial your results can be. They're closer to the mean, they have greater variability, etc. You can yield better information or more statistically significant information that way. All right, so why the pea? So again, Mendel used our common pea, or the little garden pea. Scientific name is Piscivum sativum, or Pisum sativum. And this little guy we're all familiar with, tastes very yummy. I don't think so, but other people think so. So Peas are they were and are great model organisms for Mendel to work with for many reasons. First, pea plants grow easily and very quickly. It only takes a couple weeks for them to regenerate. They produce large numbers of offspring, which are their little seeds. They are not expensive and very easy to find. And they're also a food source. So this makes them desirable to understand, right? They have some agricultural relevance that directly ties them to humans. If it has a direct connection to humans, it makes it more important and a topic that's more desired to learn about. So another thing is that pea plants can self. Now, what is self? Well, self means that they can be used to make controlled crosses. So controlled crosses, this is, well, before I get into that, pea plants, so as I said, they can self. When I say self, I mean they can self-fertilize. In other words, they produce both male and female gametes, right? Gametes are mature reproductive cells specialized for sexual fusion and containing haploid genetic content. So gametes will fuse with similar cells that originated in the opposite sex or mating type to produce a diploid gamete. So self-fertilization, this is called selfing. So when I talk about selfing, I mean self-fertilization. And they can do this because they contain, again, they produce both the male and female gametes because they contain both male and female parts. So you have something that's called the stamen, right? This is the male structure, what produces the pollen. That's these little things here, and they'll have these little tips at the ends that are called anthers. And again, that's where the pollen sits. Your bees will sit here. The pollen attaches to the little bristles on their body, and then they can carry this to another plant where it can be received by the female parts of the plant. The female parts are called the carpel. So this contains all your female structures. It's more internal, so you can see the tip of it right here, and then it goes down, and here you have your ovule. So pea plants, they can be randomly crossed, right, by any of your pollinators, but they can also be purposefully crossed with others by preventing selfing and transferring pollen manually. So this is done by removing the stamen of an individual's flower before pollen develops. So it cannot self. And then you can basically control pollination that way. So while we're on the subject, there are two types of experiments that can be performed. You have selfing, right, which is what we just talked about. Basically, you have an individual that can breed with themselves. And then there are test crosses. So test crosses are where you take essentially an individual of a known genotype or phenotype, and you cross it with, of a known phenotype but unknown genotype, and you cross it with the homozygote for that trait. And you can then determine what that genotype is by backtracking, per se. We'll get more into that in a little bit. So as I said before, one of the crucial things about pea plants that made them such a great choice of a specimen for Mendel to work with is that they had these, hello Figaro, they have dichotomous traits or binary traits. So there were 7 dichotomous traits that Mendel worked with in the little garden pea, meaning that each of them had 2 distinctive phenotypes, right? So again, for each trait, it's an either-or scenario. Now the traits were these guys, these 7 listed here, right? So don't play on my computer, don't play with the USB. Sorry. So here's the 7 traits, right? So you have flower color, seed color, and that's either yellow or green. Seed shape, it's either smooth or wrinkly. You have pod color, right? Again, green or yellow. Pod shape, it's either inflated or it's pinched. Stem height, tall or short. And flower position. For this, it's either axial versus terminal, meaning the flower grows at the top or the tip of the plant, or is growing more along the middle of the stem region. So the two possibilities for each of these character pairs, again, they're shown here, and the traits observed each came from a different gene. Right? So not only do you have these dichotomous traits, but each one is stemming from a different gene. So the gene for pod color is different from the gene for pod shape. So you have 7 different genes, each on different chromosomes. And this will bring it into importance in a bit. So Mendel began his work by ensuring that when each type of pea plant he worked with only self-fertilized for several generations. And he did this to ensure that they were true breeding with regards to the phenotype of interest. We now know, and Mendel, the genius, figured out that this meant that they were homozygous for those genes for the trait that was under examination. So true breeding plants are considered parental in transmission genetics, and this always implies homozygosity for the alleles, or the form of a gene of interest. So they can either be homozygous dominant, that would be like BB, or capital B, or they could be homozygous recessive, like little b, little b. In a true breeding plant, when selfed or self-fertilized, it will always produce progeny that are just like itself. It has the same phenotypes or traits. There's no variation, right? So you're always going to have a pure progeny derived from true breeding crosses or selfing experiments. All right, so if you are in my genetic class, you will complete these and we'll go over them in class. If you're not, you don't have to complete them. Okay, I'm not going to chase you down. Okay, so repeatedly, what he observed was that all of the F1 were identical. F1 means your filial 1, so your first generation, your first filial. F2 is your filial 2, or your second generation of offspring. So he observed that they were all identical, and that you lost this phenotype in the F1, right? So here you had an F1 that was all smooth, even though you did a cross that had crossed smooth versus wrinkled. All of them were smooth, right? All of them were this dominant phenotype. So that missing phenotype was lost in the F1, quote unquote lost, but it reappeared in the F2, which had this phenotype ratio of 3:1, with the larger proportion of the offspring possessing that phenotype that was observed in F1, and the lesser proportion of the phenotype that reappeared in F1, in the F2. So the 3:1 ratio, again, 3 is your representing the proportion of dominant phenotype to the recessive or inferior phenotype. So from these, and this is his data, right? So from this information, Mendel drew some really important conclusions. In other words, he made an experimental hypothesis, right? That's a big deal because from that he was able to stem into basically following the scientific method to formulate all these experiments and reciprocal crosses that allowed him to not only make new findings but solidify his results. So Mendel reasoned that information to create the trait was present in the F1 in the form of factors, right? Those factors we now call genes. So each gene exists in an alternative form, right? Those alternative forms are alleles, and that controls a specific trait. So true breeding strains contain identical genes. The F1 contain one of each, but since the trait is just like one of the parents rather than a mix, meaning you have one allele that's dominant, and it's masked the expression of the other trait. That other trait is recessive. And he found that the same held true with reciprocal crosses, right? So reciprocal crosses alternate sex with regard to the alleles of interest. And through this, sorry, I kind of did a shotgun method here, but through this, he devised what's called the first law of Mendelian genetics, and that's the principle of segregation. So the principle of segregation, again, an analysis of genetic crosses, when you do that, depends on understanding Mendel's two laws, hence why I'm kind of drilling it in right now. So the first law is this principle of segregation. And this law states that two members of a gene pair, or your alleles, segregate or separate from each other in the formation of gametes. Half of the gametes will carry one allele, the other half will carry the other allele. And that segregation of alleles was proven through your monohybrid crosses. Each one will get an allele randomly. When we talk about monohybrid, we're talking about just one trait. So you have this diploid myocyte, right? So you have one large R here, right? So this is the dominant. And then you have a little r allele, right? So you have one of each. Now, this diploid myocyte will go through meiosis, and it produces haploid gametes, right? So we're going through meiosis. Here's meiosis 1. Then we go through meiosis 2. They separate into their individual gametes. We have 4 haploid gametes, right? So that is the biological importance of meiosis, to make gametes, right? So you start with one pair, right? You go through meiosis. Meiosis produces 4 gametes. Each gamete is haploid. One member of each of those pairs, right? So one member of this chromosomal pair, one member of this chromosomal pair. One of each is going to end up in one gamete. So each gamete is haploid. And here we're just assuming that the first will be identical. It doesn't really matter. But anyway, 2 will have your big R allele. 2 will have the little r allele. And these are what go on the axes of your Punnett squares. As soon as I say Punnett squares, I feel like an entire room just, their soul dies. But they're not bad. Some people have a difficult time understanding them. So hopefully I can help alleviate some fears that you might have. But trust me, Punnett squares are great, right? So Punnett squares model a biological reality, per se, by modeling possible fertilization events. So, for example, you make a square for, let's say we're doing a monohybrid cross here, meaning we're following one gene. So let's have a square that has axes of A, big A, and little a, and then big A, little a. So you'll have big A, little a on your horizontal axes or your y-axis, and then big A, little a on your on your x-axis, and then big A, little a on your y-axis. When you complete that, you end up getting a 50% chance for each gamete at each axis, right? So the combinations inside the square represent the predicted offspring based on the gametes obtained from sperm or eggs. And what you'll end up getting is big A, 2 big A, little a's, and then 1 little a. So you get a 75% chance, or 3/4 chance, of a dominant phenotype, meaning your phenotypic ratio is 3:1. The genotypic ratio is 1:2:1, and that's for a monohybrid cross. Again, monohybrid, you're following one trait of interest and essentially mating a true breeding parental plant that show the opposing phenotype for the trait of interest. So you're taking up, in this example, true breeding parent for the purple flowers and a true breeding parent for the white flowers and crossing them to get F1 generation. So some terminology. So parental generation is the P generation. It's the true breeding generation. So true breeding, again, that means it's homozygous for the allele of interest. In this case, we have big S crossed with little s. This is the recessive, this is the dominant. Now, the progeny of your P generation is called the first filial generation, right? So, as I said before, the filial 1. So that's designated as F1. This is your F1 generation. Now, these offspring will be hybrids, right? So, when you hear hybrids, that's referring to offspring from different true breeding organisms. So in this case, your F1 generation would be this little hybrid of your parental cross. So it would be big S, little s. That's what its genotype would be. Now, when the F1 or your filial 1 is interbred or selfed, you get the second filial generation, or F2. Right? So you take an F1, you cross it with another F1, you interbreed them, you get your F2 generation. And then subsequent interbreeding events will produce your filial 3, filial 4, filial 5, or your third, your fourth, your fifth offspring generations. So here we have a monohybrid cross. Here's another monohybrid cross. This is looking also at wrinkled or smooth. It's just using R instead of S. As I said before, reciprocal crosses. So that alternates the sex with regard to allele of interest. I'm trying to think if there were any other important terms I mentioned. So with this example, Mendel observed that all of your F1 generation were smooth, right? And we now know that this means they all have the genotype big S, little s. So this is what I mean by the principle of uniformity in the F1. This is what another important concept that Mendel found. So this is when you have just the dominant trait appearing in the F1 generation. You essentially lose this recessive trait, but it reappears in the F2 generation, right? This is the that trait that I said, quote unquote, is lost. So with Mendel, he continued to allow the F1 from this cross here to self, right? So he allowed them to self-fertilize or interbreed. And what he noticed was that the little wrinkled P phenotype reappeared in the second filial, or your second generation, your F2. He also noticed that in this cross and in similar crosses, when he examined the F2, he always repeatedly observed the reappearance of phenotypes that had disappeared in this F1. And that's this notion that kind of forms the principle of uniformity in the F1. The loss of the recessive phenotype in the F1, and then it reappears in your second generation. So note that we're going to move into dihybrid crosses, right? So monohybrid crosses, again, you're tracking one trait of interest. With dihybrid, you're looking at two, or you're looking at two genes of interest that bear different phenotypes. So note that there are two ways of doing a dihybrid cross for the same traits or genes, and that's what's shown here. A common student misperception is that one parent will be homozygous dominant for both gene pairs of interest, and the other homozygous recessive. And that's largely because they don't truly understand what parental means. Parental just means, again, homozygous for that gene of interest. It does not dictate whether you are homozygous dominant or homozygous recessive. So you can have a parental generation that is homozygous dominant for both gene pairs, or homozygous recessive for both, or homozygous dominant for one, homozygous recessive for the other. It doesn't matter. Again, parental just means it's homozygous for the genes of interest. It doesn't dictate whether it's all homozygous dominant or homozygous recessive. So you can do a dihybrid cross, again, with, in this case, you can do it with big Y, little r, or little r, and little y, big R. It doesn't matter. Your Y's are one gene, R's are the other. So here, this is homozygous dominant, this one's homozygous recessive. It doesn't have to always be this way. You can have one of these be, well, for the parental generation, it should be this way. But if you were just doing a random dihybrid cross, it doesn't have to be specifically dominant for both or recessive for both. It can be dominant for one and recessive for the other. So Mendel's hypothesis, kind of starting here, was along the lines of what's called independent assortment. And his actual results definitely support this hypothesis that he formed. Now remember each of the dichotomous traits that Mendel investigated. Each of these were on different genes. Now because the two genes what are being shown here for seed color and seed shape, since they're not on the same chromosome, the chance of inheriting either a big R or a little r does not affect the chance of inheriting a big Y or little y. That's independent assortment, right? So we go through, make our Punnett square. While you do your little cross, tend to go through meiosis, right? So now we're in the filial 2. So you have your 4 haploid gametes, right? You have your this guy, big Y, big R. You have big Y, little r. Right? They're all different possibilities stemming from here. So you could also have little y, big R. That's this gamete here. Or you can have little y and little r. Right? That's this 4th gamete. Each will have equal proportions because they're on different chromosomes. So each has an equal chance of occurring or being present in the gamete, and that equal abundance is 25% or a quarter. And that's because of random assortment of chromosomes and how they assemble during division. Now, this can only happen if those two genes are on different chromosomes. If they are on the same chromosome, you actually have to map out the distance between those genes because the genes might be linked. And that goes into a different topic, which we'll discuss a few lectures from now. So this is Mendel's, where this moves into Mendel's second law, the principle of independent assortment. So this is the theory that two alleles of two genes pass into gametes without influencing each other. So the formation of random combinations of chromosomes in meiosis and of genes on different pairs of homologous chromosomes by the passage basically accords to the laws of probability of one of each diploid pair of homologous chromosomes, and they will pass through into each gamete independent of each other pair. So if independent assortment is happening, we expect to see a phenotypic ratio of 9 to 3 to 3 to 1. Again, that's if they're not on the same chromosome. If they are, this will change those phenotypic ratios. But if they're not on the same chromosome, again, we expect to see a phenotypic ratio of 9 to 3 to 3 to 1. Okay, so let's do a little example here. So if two genes, let's say, what do we have here? Big A and big B are close together on the same chromosome, they do not assort independently and are said to be linked, right? This is when recombination occurs, genetic recombination. Whee! So you start with a diploid myocyte that has two non-sister chromatids, right? One with big A and big B, as shown here, and the other with little a, little b. Now the chromatids basically stick together and they swap alleles, right? And this can occur at multiple locations. So you get 4 haploid gametes that are produced. 2 will be non-recombinant gametes. The other 2 will be recombinant, right? So your non-recombinant are, or your non-crossover, are your big A, big B, and little a, little b, right? So these are your 2 non-recombinant gametes. And then you have your 2 recombinant gametes, right? So these are big A, little b, and little a, big B. So again, your non-recombinant gametes and your recombinant gametes. Now, the DNA sequence of 2 sister chromatids is completely identical apart from some very rare DNA copying errors. Non-sister chromatids, on the other hand, what I was talking about here, these guys, these refer to either one of 2 chromatids of paired homologous chromosomes. That is the pairing of a paternal chromosome and a maternal chromosome. So when we have genes that are located on different chromosomes, right, like the ones that we were talking about right before we got to this slide, those will assort independently, right? And they have a recombination frequency of 50%, right? On the other hand, linked genes have a recombination frequency that is less than 50%. The gametes, however, occur at unequal frequencies, depending on the locations of the chromosomes and the distance between the genes. If they're far apart, there will be a higher amount or frequency of the recombinant gametes. So if these were far apart from each other, you'd see more of your recombinant gametes, or more of these crossover gametes present, a higher percentage of them in the offspring. If they're not far apart on the chromosome, if they're closer together, you'll have more of these guys, your non-recombinant gametes. So in other words, the actual ratios will be different if A and B are on different chromosomes and will be different depending on the distance between them on that chromosome. If they're on the same chromosome and based on the distance between them. So this all goes into an advantage with sexual reproduction for eukaryotes. During sexual reproduction, the genetic material of two individuals is combined to produce genetically diverse offspring that differ from their parents but also bear some similarity. Now, the fact that most eukaryotes reproduce sexually is evidence of its evolutionary success. In many animals, it's actually the only mode of reproduction. So during prophase 1 of meiosis, our cells undergo crossing over or recombination, right? So this is very important because it shuffles genes on the same chromosome, and that allows for some genetic diversity to occur. Genetic diversity is great. You need some amount of genetic diversity to maintain fitness in the realm of stochastic events or stochastic change. So what ends up happening is that you have this gene exchange between homologs. And the gametes are a little different from the myocytes. And again, this is what introduces genetic variability and also allows for different phenotypes. So there's also another mechanism for introducing genetic variability during or in sexual reproduction, right? It's not just crossing over or recombination, but there's a second way, right? So the second way is, well, independent assortment, right? You have this random alignment along a plate, and then you can shuffle chromosomes between those pairs, and voila, you end up with introducing genetic variability. Okay, so sorry for the little deviation there. I think it was pretty important. Anyway, so let's return now to Mendel's principle of segregation. Like all good scientists, once Mendel devised his hypothesis, he sought as many ways as possible to test it. The classical method he developed was this thing called a test cross. Now, test cross, as I said before, it involves an organism with a dominant phenotype but unknown genotype. So, for example, you have something that's big B, or big B, little b, but we don't know that. We just know it has the dominant phenotype. So you take that organism and you cross it with a homozygous recessive organism for that gene of interest. Now, by examining the results of the cross, you can determine the genotype of the individual having the dominant phenotype, as shown here. So it's essentially this way to verify that the organism of interest is, in fact, heterozygous. And when I'll use a term frequently called zygosity. That's what this is used for, to determine zygosity by analyzing the proportions of your offspring phenotypes. Now, zygosity can either be heterozygous or homozygous. Again, here, what we're looking for is to either verify that the organism of interest is heterozygous or to kind of work our way backwards and determine the genotype of that individual having the dominant phenotype. Okay, so let's do out a little example here. So, for example, let's say the individual in question is homozygous dominant, right? Let's say it's big P in this case. Now, you test cross it with a homozygous recessive individual. That's this guy here, big P, but it's white. Sorry, little p, but it's white. Now, results would give you offspring that are all purple and heterozygous, right? 100% purple. There are no recessive phenotypes among the offspring. If the individual in question was instead heterozygous, meaning their genotype was big P, little p, instead of big P, you have big P, little p, the result would be 50-50, right? The offspring, 50% would be purple, or you're heterozygous. The other 50 would be white. They're homozygous recessive. So a test cross of big P, little p with little p yields a phenotypic ratio of 1:1, right? 50-50. 50 purple, 50 white. Half purple, half white. So if the test cross yields a resulting phenotype of 1:1, the parent is heterozygous. So again, you look at the phenotypic ratios of the offspring to determine the genotype of the parent in question. So again, this is for my genetics class. You guys can work on this and we'll discuss it in class, most likely on Thursday. All right, so moving into a real life example. Real life examples are great because they kind of bring everything more to earth, right? You don't have this abstract thought. You can actually kind of start placing your knowledge into real life examples. So our real life example is albinism, or albinism, however you want to pronounce it. Specifically, oculocutaneous albinism. So this refers to a group of inherited disorders that are characterized by little or no melanin production. Melanin is pigment, right? It's the pigmentation of your skin, your hair, and your eyes. So affected individuals typically have very fair skin, like this individual, this young man here, or white or very light colored hair. They have high risks that will dealing with sun exposure, and that greatly increases their risk, I should say, of skin damage, cancers, including melanoma, and also reduces pigmentation in the colored part of the eye. That's called the iris, and the light sensitive tissues at the back of the eye. That's your retina. So as a result, these individuals usually have vision issues, right? So these include photophobia, which is increased sensitivity to light. Also something called nystagmus. That's this rapid involuntary eye movement. Now, oculocutaneous albinism, or just albinism, is inherited in an autosomal recessive pattern. So that means both copies of a gene in each cell have mutations. Most often, the parents of an individual with an autosomal recessive condition each carry one copy of the mutated gene, but they do not show signs or symptoms of the condition because it's recessive. So the actual gene that's associated here is your OCA1, or your OCA1 gene, and this is associated with abnormalities or mutations in, OCA1 is the type. You have type 1, type 2, and type 3. Might go to 4, but I know it goes to 3. Anyway, your type 1 is associated with abnormalities or mutations in the tyrosinase, or your TYR gene. And the TYR gene is responsible for the production of the enzyme tyrosinase, right? And this is a key enzyme in the formation of the pigment melanin. In melanocytes, tyrosinase oxidizes tyrosin into dopa, 3,4-dihydroxyphenylalanine. That's a long name for it. So it will oxidize that into dopaconone. Now, some TYR mutations result in the production of a completely non-functioning tyrosinase enzyme, thus no melanin pigment is formed. This results in just a slightly different subtype of your type 1 oculocutaneous albinism called OCA1A. For OCA1B, you have a slightly different mutation of the TYR gene. In the case for OCA1B, the tyrosinase enzyme has limited enzymatic function, but is still able to produce small amounts of the melanin pigment. And in that case, you'll have the actual accumulation of the pigment in time in the skin, the hair, and the eyes. This gives you some kind of real-world examples to tie in everything that we've talked about. You can take, now the reason why I'm talking about this is because we're going to use it in the next exercise to look at how you can, basically the inheritance of recessive traits or recessive disorders, in this case, albinism. So work through these. Again, we'll go through them on class on Thursday. If you are not in my class, you are more than welcome to go through these exercises. There's nothing stopping you. But as for now, I think that is all I wanted to talk to you about. So have a great evening. I'm sorry for all the squeaking from Figaro. He means no harm.